Alright, so today we're going to start our topic of 2.6, the nucleic acids. So at the start of topic 2, we have talked a lot about the different kinds of biomolecules. They come in the flavors of the carbohydrates, the proteins, and the lipids. And so now today we're going to focus on the nucleic acids. And so there are basically two kinds of nucleic acids that we're going to talk about. The DNA, which you've probably seen before, and the RNA are the two members of the nucleic acid family that we're going to talk about. But let's take a little, tour down the history road to figure out how these were first discovered. So the microscope was not invented until about 1590 by this Zacharias Janssen person. And later, Robert Brown was looking at the structures of cells under the microscope. And then that's when they saw this kind of structure showing up in different kinds of species in the center that was present in both plant and animal cells. And because they thought, hey, it looks like a nut, let's call it a nucleus, because nucleus means like little nut. So all they knew was that we had this structure in the center of cells that was pretty prominent, but they didn't really know what it was or what it was for. So later, kind of a lot later, we have the isolation of the nucleic acids for the first time by Friedrich Miescher in 1868. And so what he was able to do is he took the nucleuses out of white blood cells, and he figures out that it's not lipids because he digests it with lipase and the material didn't degrade. He knew it wasn't protein because when he put the proteinase enzyme in there, the material also didn't degrade. And he tried a bunch of different carbohydrate enzymes, and because that didn't degrade, he knew that he had a new class of biomolecules because none of these enzymes were able to chew them up. And remember, enzymes are specific. They can only degrade certain kinds of materials. So because of where it was located, he says, okay, we're going to call this nucleic acids. He did note that it increased in mass prior to cell division, and he could also tell that it had a large amount of phosphorus because they were able to do those techniques. But apart from that, they didn't really know what it was or what it was for until much later. So now we know that DNA is the molecule of inheritance. We know that it contains a large amount of carbon and hydrogens. It does have some oxygen as well as nitrogen. And again, they were able to figure out that it had phosphorus. And as far as our biomolecules are concerned, this is the only biomolecule that has phosphorus. The other three do not. So most of our biomolecules, except for lipids, have some kind of monomer, right? So the carbohydrates, the monomer of the polysaccharides was the monosaccharide because we take one monosaccharide and string them together through condensation reactions and build a large polysaccharides. When we were talking about the proteins are just amino acids. So the monomer of the protein is the amino acid, and so we can string the amino acids together again through condensation reactions to create the long proteins. So just like the monomer of polysaccharides is a monosaccharide and the monomer of a protein is the amino acid, the monomer of a nucleic acid is this nucleotide. So this whole structure here is the monomer of both DNA and RNA, and it comes in three parts. We've got a phosphate group, a pentose sugar, and a nitrogenous base. So, okay, the whole thing is a nucleotide, and this is going to be the thing that repeats that we can join together to make nucleic acids. So we have our phosphate, which is attached on that 5' carbon to a pentose sugar, and then on the 1' carbon we have some sort of nitrogenous base. So this is how we draw it for DP purposes, with the phosphate as a circle connected to a pentose sugar, and again just the square for the nitrogenous base. You don't have to do anything more complicated than that. So again, nucleic acids are just polymers of nucleotides. So this whole thing is a nucleotide, and it can be joined to another nucleotide. Again, these are condensation reactions, so when they join, you produce that water molecule. So, like many of our other biomolecules, the nucleotide sugar has directions and numbers attached to it. So remember, for our carbon labeling rules, we start with carbon 1, 2, 3, 4, and 5. So same thing down here, we have carbon 1, 2, 3, 4, and 5. So the convention for the nucleotide is we consider the 5' to kind of be the top and the 3' to kind of be like the foot. Like if we go back to our pizza man example, the 5' would be the top, he's holding up the pizza, and then the 3' would be like the foot. And when we are adding a new nucleotide to the polymer, and again, that's occurring as a condensation reaction, it will only attach to the 3' carbon. So you are going to attach this 5' to the 3'. And when we're looking at this chain, right, which part of the nucleotide is different here? So we've got 3 parts. We've got phosphate, we've got the pentose sugar, and we've got a base. And so if you look at the nucleotide, 2 of the parts repeat, but one of them is different. So the part that repeats always is the phosphate and that pentose sugar. And what is changing is this base. So later, when we're talking about sequences and DNA sequences, you'll get letters like A's, G's, T's, and C's. And so this is the part of the sequence that is being read. That's where those letters are going to come from. And later, you're also going to see that there are 2 strands of DNA and that the direction matters here. And so the 5', 3', what we're talking about is the number of these specific carbons. That's where that convention is coming from. So when we're making polymers of nucleic acids, we only add to the 3' direction. Those are occurring in condensation reactions. And again, it's the bases that change. The phosphate sugar stays the same. So both DNA and RNA are polymers of these nucleotides. And again, we're only adding a new nucleotide to the 3' end, and it's these bases that change. But there are some differences between DNA and RNA. And so not just the first letter, although it is related. So you've probably seen the double helix before for DNA. So DNA is double-stranded. And if DNA is double-stranded, it means that RNA is single-stranded. And let's talk about the bases. So for DNA and RNA, there are both 4. So for DNA, it is A. And again, just the sequence is this part here is going to be the nucleus. We have bases of A, G, C, and T, which stand for adenine, guanine, cytosine, and thymine. So these are the bases that we will find in DNA. RNA has something really similar. It also has A's, G's, C's, but now instead of a T, it has a U. So for both DNA and RNA, we have adenine, guanine, and cytosine. So these are shared. This is the same between the 2 of them. But what's different is that thymine is only found in DNA. So if you see a sequence and it's got a T in it, it's automatically a DNA. But if it's RNA, it will have uracil. So the basis is one difference here, so is the structure. The other has to do with the sugars. So DNA stands for deoxyribose. And so if you've been with the DP program for a while, we have been drawing the ribose, the pizza man, for kind of a while. We know that it has its 2 feet, its 2 arms in the pizza. So ribose is the sugar in RNA because it is ribonucleic acid. So similarly, the sugar of DNA is deoxyribose. The only difference is that it's deoxyed. It's missing an oxygen right here. So DNA will have the deoxyribose in this part of the nucleotide, and RNA will have ribose in that part of the nucleotide. Again, the only difference between them really is this OH. So let's look at the structure of DNA. So this graph is a little incorrect because we don't draw hydrogen bonds with solid lines. They should be dotted, but let's go back to our nucleotide. We have our phosphate here. We've got our deoxyribose if we're talking DNA, and then we have our bases in the center. And the bases are going to form hydrogen bonds, and they're going to form base pairs. So again, don't look at this here when you draw them. Make sure it's dotted lines because hydrogen bonds are not solid. This implies a covalent bond, which is incorrect. So hydrogen bonds are forming between this set of bases, and any solid line that you have here is going to be a covalent bond. The other thing that you may have noticed is that the strands on this side are kind of top right side up, and the strands on the other side are kind of upside down. So this is going back to the direction that on the left side, this is going 5', because again, we're talking about the carbons that are numbered. If we look at that number again, it's 1, carbon 2, carbon 3, carbon 4, and we're going to pretend you can see carbon 5 here. So the top on this side is carbon 5, and then the bottom on this side is 3', because it's 1, 2, 3, carbon 3. So this side it's 5' at the start, and 3' on the bottom. We call this the sense strand, the positive strand. And whenever you have a DNA sequence, on the sense strand, it'll be like CAGTC. That's how you would pull the sequence off. And anytime you see a sequence, if it's not specifying, it's probably from the sense strand, from that positive strand that's going 5' to C'. On the other side, it's attached to a complementary strand. And on this side, the deoxyribose is flipped upside down, and the direction is opposite because, again, we're going carbon 1, carbon 2, carbon 3. So this would be 3' to 5' on this side. And so we call this one the negative strand, or the antisense strand. And so you're probably noticing here also that, we're forming bonds in between them. So we'll talk in a second about the base pairing rules. But again, just take a look at this, that we have nucleotides that are repeating, again, that were joined in those condensation reactions. What is changing is the bases, but the phosphate and the sugar are really the part that's repeating. It never changes. It's only the bases in between that are changing. So again, we have our phosphates, we've got our sugars. If it's DNA, this is deoxyribose, and we've got our bases with the hydrogen bonds in the center. When you see the conventional diagram of DNA, it's usually this spiral with this sort of like lines in between them. The bases are in between there, and they're forming their hydrogen bonds. So if we look at a different diagram, this would be the double helix ribbon that you've probably seen before. And what's tucked in here is the phosphates and the sugar. That's why we say that DNA has a sugar phosphate backbone, because the ribbons are implying the sugars and the phosphates that are repeating. And again, it's the bases that are reaching out in the center and forming those hydrogen bonds. So they were just sort of simplified as lines, but this is really what's happening in between them. And what you might have noticed is that only certain pairs are forming. So there are some rules about DNA base pairing. So A will always pair with T. So you can think of it as like apples in the trees. And G will always pair with C. So you can think of that as like cars in the garage. So apples in the trees and cars in the garage. The other thing to keep in mind is that the AT base pairs will only form 2 hydrogen bonds, whereas the GC base pairs will form 3 hydrogen bonds. And this diagram is correct in that we have the dotted lines denoting those hydrogen bonds, and the number is accurate. So later when we talk about like PCR, know that the AT sections are going to melt faster because there's fewer hydrogen bonds between them. So when we're drawing our DNA molecules, so say I ask you for a base pair, what I want you to do is start with a nucleotide, which again has 3 parts. So I'll have my phosphate, which we're going to just represent by circle. And we're going to connect that to our pentose sugar, which if it's DNA, it's deoxyribose, and please label them like I have it here. And then you're going to attach the base to one side. And so A, remember, pairs with T. And again, we're forming those 2 hydrogen bonds. Please don't draw covalent. Make sure you have that dotted line. And we're going to go ahead and draw our complementary nucleotide on the negative strand, anti-parallel, so this is correct. And so A pairs with T, and then G is going to pair with C because it's apples in the trees and cars in the garage. And again, the GC pair has the 3 hydrogen bonds. And remember that when we're adding nucleotides, we always add it only to the 3' carbon, so it's always a 5' to 3', 5' to 3', and that's just going to repeat on both sides. So this is what we need to know for topic 2.6. Be able to tell me the difference between the structures of DNA and RNA, know which bases belong with which nucleic acid, know which sugar belongs with which nucleic acid, and be able to draw these base pairs. So that's it for 2.6.